Yeah, as Janice mentioned today, I'm going to be talking about something a little bit different than most of the topics we've been hearing about so far. And that's the idea of whether or not Phragmites is hybridizing. And when I say Phragmites hybridizing, I mean hybrids between the native and the introduced Phragmites in North America. So this is a project that was really, it came from a kind of ground up initiative facilitated largely by Janice. So thank you for that, Janice. And really what happened was a bunch of on the ground conservation manager people were noticing something funny going on with Phragmites. They would see stands of Phragmites that appeared to be, in their eyes, native Phragmites, but the way they were growing appeared to be more dominant than they expected. Now, you know that Phragmites and native Phragmites often grow together. They are wind pollinated, so there should be no barriers to one type pollinating the other. And so the question arises, are they in fact creating hybrids? Next slide, please. Next slide, please. There might be a bit of a delay. And we have previously detected hybrids, but rarely. So one study found 5 hybrids in Ontario after surveying a range of sites, including some at Long Point, where the native and invasive do co-occur. Another study found 1 hybrid in upstate New York. Next, please. The exception here is Las Vegas. There's many hybrids in Las Vegas, in regions around Las Vegas, and they are, in fact, behaving in a rather invasive manner. So this is a worry, because if we think about things like Typha in the Great Lakes region, it's the hybrid Typha x glauca that is the invasive problematic one. And so there's obviously some concern that if we sometimes start with low frequencies of hybrids, but they can become more common and over time become increasingly invasive. So people are concerned to monitor it for that reason. The other reason people are concerned to monitor it, of course, is, and this has been touched on by a number of other presentations, if they see a stand and they want to control it because they think it's invasive, but it is in fact native, that would be counterproductive because, as other people have talked about, there's the importance of maintaining genetic diversity of the native populations. Next, please. Okay, next slide, please. Or next point. Yeah, so as I mentioned, the hybrids around the Las Vegas area do seem to be becoming more invasive. They have higher stem and panicle densities. They have higher seed output than native, even though the germination rates are lower. Have higher rhizome viability, higher genetic diversity. Next, please. Yeah, sorry, keep going. I've already touched on these points. It's a little bit weird not controlling my slides. So from this study, we got a bunch of samples from different regions, different managers who had independently observed these unusual growth patterns in these plants. And we identified a few potential outcomes. So what could be happening is that these are, in fact, hybrids. So they have morphology that appears like natives, or it's maybe somewhat intermediate, but growing invasively, maybe they are hybrids. Next. Yes, a concern. The second potential outcome is that they have a wider range of morphological characters in invasive plants than previously documented. So plants that people think are native are in fact invasive. The third potential outcome is that the native plants have more diverse growth patterns than previously documented. So what looks like unusual growth pattern in native plants is actually quite normal. And next, please. Native plants may be becoming increasingly dominant. And that, there is evidence of this having happened before. As environments change, then sometimes native plants can change their growth patterns. Next slide, please. So these are just a few photos that were sent in just to show examples of the types of things people are observing. These were sent in by Judith Jones. Just next slide. It's just showing that they are native appearing plants, but they seem to be, next please, more, I guess, growing in patterns that people did not expect to see them. Next, please. And next one. And this map just shows the different regions from which we received samples. So you can see it's spanning a range of different types of parks, habitats, different conservation authorities, and so on. Next, please. And as people collected samples for us to do the DNA analysis, we also asked them to look at some morphological characteristics. And these are just characteristics that previous studies have identified as being useful to differentiate native and invasive plants. And I'm sure you're all familiar with these characteristics. And I'm sure you use them when you're looking at the Phragmites. So this just shows the characteristics that people recorded. Next, please. In order to determine whether they were native, invasive, or hybrid, we genotyped 127 plants from these quote-unquote questionable stands, the ones that had people scratching their heads a bit. We genotyped them on the basis of 7 microsatellite loci, which can be used to infer whether they're likely to be native, invasive, or hybrids, because we have a whole bunch of data from earlier studies of known native and known invasive, and we could compare the data sets. We found that the large majority of samples that had been sent to us were in fact from native Phragmites. A few of them were from introduced. We didn't find any evidence for any hybrids in the mix. Next, please. This just shows the distribution. Again, just reiterating, you can see that most of them are native. There's a few on Manitoulin that were introduced. Next, please. We compared our results to the data that people recorded in the field based on morphological characteristics, and we didn't find as tight a correlation between the two as one might hope. It's possible that there's regional variation in the morphological characteristics, and depending on where you are, maybe some will be more useful than others, because the studies that have come before were based on fairly limited geographical regions. We did have missing data in a lot of places. Not, for example, some of the sites that were visited were hard to access, and if people were there prior to the flowering season, they weren't always able to go back when the plants were in flower. We also know whenever we're looking at measurements, you do get inter-observer differences. And although we did ask for multiple measurements on things like gloom length, repeatability can be challenging, particularly in field work, particularly when you're doing small measurements. It's also possible that there was subconscious bias, people having maybe expectations about what these plants represented. Next, please. Overall, we found that the characteristics most useful in identifying native Phragmites were stem color, stem texture, and upper gloom length. And you can see none of these are hitting 100%. So what that means is in no case was a plant that we identified as native on the basis of molecular genetic data consistently identified as native on the basis of a particular morphological character. Next, please. Characteristics most useful in identifying the invasive Phragmites, so that would be stem flexibility, leaf color. We didn't have any data on gloom lengths, and you'll recall that our sample size of the invasive was very small. So I take this one with a bit of pinch of salt, but again, we're not seeing any characteristics that 100% of the time was able to identify invasive Phragmites. Next, please. Interestingly, on the form, we had included categories of stem density and maximum height of the stands. And most of these were in the what's really been identified as a normal range, an accepted range for native Phragmites, which to me suggests that maybe when people are recording these stands that seem to be growing invasively but look native, I think maybe a little bit more investigation, maybe a bit more documentation about the stands would be useful. This is one thing that it was a little bit difficult for me to get information from, was asking people to describe the stands in a way that could be, using types of measurements that could be reproduced. Next, please. Okay, so coming back to the potential outcomes, we're crossing off number one. We know that they're not hybrids. And we're also crossing off number two because most of them were not invasive. So this leaves us with two possibilities. One, these native plants have more diverse growth patterns than has previously been documented in North America. Or two, native plants are becoming increasingly dominant. So let's just look at a little bit more detail about some of these two options. Next, please. I actually, going back a few years now, did a study on Phragmites growing in the UK, where it's native. And reedbeds are actually a priority habitat in the UK Biodiversity Action Plan. So it's by no means a dominant or invasive species in the UK. Next, please. So this map just shows the sites that I looked at. And one thing that surprised me about looking at these sites around the UK is the range of habitats that Phragmites can tolerate there. So sometimes very, very low pH, as you see at this peat bog here, very low pH. It can tolerate brackish conditions, saline conditions, and of course, fresh water. It can tolerate disturbed sites. Sometimes it's found in very small patches, intermingled with lots of other species. And at times, which I think is more consistent with expectations here of native Phragmites, at times it can grow in very expansive patches. Next, please. So here's just a few slides illustrating next just how it's growing, even though it's native and it's considered, you know, quote unquote, well behaved there. It does grow in unusual. See how this is an incredible monoculture here. This is actually in a brackish habitat. It's just, so this is the native Phragmites in the UK, which is the invasive one here, but it's not considered invasive here, but you can see it's very, very dominant of this particular site. Next slide, please. And another one here adjacent to an agricultural field, just a huge reed bed. So really, really diverse types of growing conditions. And this was a review published a few years ago, a couple of years ago, I can't remember exactly when, on sort of a review paper of Phragmites in the UK. And they did point out that, and even though it comprises protected habitats, it is sometimes considered a problematic high-performing species because of these dense monospecific populations that it can create that require active management. So this is interesting because it raised the question that maybe native Phragmites in North America also can, you know, follow this kind of framework. Next slide, please. This is just, so scenario 4, growth patterns of native Phragmites are changing. This is, however, another thing that we cannot rule out. And this is perhaps a more consistent conclusion if we look at the inter-year observations by land managers, conservation managers. And just as a very brief aside, I can't even remember how we started this conversation, but I had some email conversation with Rosemary Curley, who works for the PEI Invasive Species Council, and she independently had identified the same thing, what she thought were native stands that seem to be becoming increasingly dominant. Next slide, please. So it does raise the question, oh, this was just from a map that she sent me. We don't need to worry about the details of the map. It just illustrates different ways in which she has been documenting these stands, both through aerial imagery and on-the-ground imagery. So it does raise the question that maybe through some sort of environmental changes, the native plants are starting to grow more dominantly. Important caveat, just because we didn't find any hybrids at the moment does not mean that there will not be any hybrids in the future. So think of Las Vegas. And hybrids, if we look at, as I mentioned earlier, if we look at the typhus situation, are a potential concern. So here's my breaking news that actually is came from an email from one of my collaborators at the weekend. So I've been collaborating with a couple of people in the States. One of the obstacles we have at the moment is determining whether Phragmites are hybrids. So it's pretty convoluted, time-consuming process right now. Also, they're polyploid, which makes them a pain in the butt to work with for doing genetic work. So this has limited, certainly, the number of samples that I've been able to test for people. But I believe, and this is still in development, but we seem to be on the cusp. I think we have actually developed a much, much faster genetic test that can be used in the lab to identify hybrids. And this will certainly make it much easier to do just a very quick, you know, within one day, find out if these are hybrids or not, as long as there's a, you know, person to do the work and the consumables are present. So I think this is going to make it much easier to monitor. And if the native ones are growing more expansively, that might also increase the likelihood of hybrids arising. So that's certainly something to keep in mind. Next slide, please. Do I have another slide? Or is this the last one? Oh, right, of course, acknowledgements. So conclusion, there's multiple independent observations in Ontario, actually also in, I forgot to mention, in Michigan, these observations have been made, and as I did mention, in PEI. So there does seem to be something going on here. They're not hybrids, but there seems to be something going on with the native species. Certainly ongoing monitoring is probably going to be important for this. And here are acknowledgements of various people who had provided information about this, sent in samples, and so on. Okay, any questions?
There we go. I'm going to direct you to the Q&A panel. It looks like quite a few questions came in during your chat.
Okay, so is there an up-to-date map that shows the distribution of native Phragmites in Ontario? Not to my knowledge, and that actually would be really useful. Where can we get DNA testing done to determine if it's native or invasive? So I don't have any students working on it right now. There is a lab at Trent that can probably do it. As I said, it does currently take a while to do it. I think if you are interested in this and you get back to me before the growing season, we'll have sorted out this new method by then that I think can be done very quickly. And so if you are interested in getting this done, send me an email, not today, because I'll just have lost it by then, but maybe sometime in March when you're sort of starting to plan your field work for the summer and so forth, and we can talk at that point. I don't know, I can't say at this point whether I could do it in my lab, but if not, I know a lab that I think would be able to do it, particularly with this faster method. It's really interesting that the morphology isn't 100% diagnostic. Yeah, and I think one of the challenges is I don't think that the morphological characteristics and publications that describe the different, you know, invasive versus native were wrong. It's just that those measurements were done under such controlled conditions, and they were done really carefully with repeatability. It wasn't a whole bunch of different people measuring them. It wasn't people trying to do them while they're doing field work and doing 100 other things at the same time. So I think what this is showing is not necessarily that the morphological characteristics are not reliable, but maybe more that they're difficult to record reliably in the field. And so that's something else to be aware of. Okay, next question. We hear this. How many haplotypes of native frag are there? There's a few, and every now and then a new one pops up, but I don't know the answer to that because the native ones have been studied much less than the invasive ones. And they have sort of been the poor cousin. And as I mentioned, you know, one of the things we might be seeing here is a big knowledge gap. And so I think it would be really interesting actually to spend more time studying the native stands and getting to understand them a little bit better and actually seeing how much diversity, how much variation is there, both genetic variation, variation in growth patterns, you know, ecological adaptations and so on. Any research on generation of frag from seed? Oh, okay. Well, Verena, who's a PhD student who is in the audience today, she has tried to germinate them. We have been able to germinate them in the past. She tried for quite a while and had no success. So again, knowledge gaps around this. Why can we sometimes germinate them? Why can't we? Hasn't been a ton of research on that. If hybrids so rare on these separate species, that's actually a really good question. One of them is, so americanus is considered a separate subspecies, but it's a really good point. And it's kind of surprising that they're not hybridizing more. So why they're not hybridizing more, I have no idea. There might be variation. It could be something really simple like variation in the time at which they produce pollen. So just, a slight a reproductive barrier in timing of reproduction. And that's certainly something that could be quite fluid if we see environmental changes. But yeah, really don't know. And that would be another interesting area of research because as we know, hybridization is so common in plants. Why are they not hybridizing more? They're also polyploid though, so that does confuse things a little bit more. What keeps the native under control? What keeps any native under control? Does something eat it? Yeah, things eat it. But I mean, that's just evolution, isn't it? If all native species were dominant, we wouldn't have stable ecosystems. So I think that's kind of a much broader question. Any gene flow happening from non-natives to make the natives more aggressive? Well, if that was true, so I think what you're describing there is introgression following hybridization, but the evidence for hybridization other than around Las Vegas is so far very, very low. So one thing I would really like to find out is the pollen coming together? So are the barriers to hybridization pre-reproductive or post-reproductive? Is it something to do with the polyploid state and it's just, I don't know, something's incompatible there at the genetic level, or is it something it's just, which would be good news because that means we should be less likely to see hybrids ramp up, or is it something around timing or of pollen dispersal? Because they do grow very close to each other, and the pollen should be blowing back and forth unless the timing is quite different. And I'm not aware of anybody actually having studied that, so I think it would be worth investigating a little bit more. It's something to think about. It would be kind of a neat study to see when pollen is being released.
Thank you so much. There's so many questions. So awesome
So many questions. Yeah, we're just at the tip of the iceberg
It
here.
really is. We should have, I don't know, days of fragging. There's just so much, and it's thank you so much for that presentation. Yeah, there's a lot there. You said a lot of questions there, but you helped answer some of them for me, and I appreciate that. I have a lot more from you. Are you going to continue on with this research, Joanna?
I don't know. I don't know. I mean, as you know, I do. Typha is more the interesting one because they do hybridize. So for an evolutionary biologist, that makes them more interesting. But it is, the more I think about it, it is really interesting why we are not seeing hybrids in Phragmites. But of course, it's harder to investigate why something is not happening than to investigate why it is happening. But to me, apart from anything else, what this shows is, I think as a community, we have maybe made a little bit of an error in that we are studying invasive Phragmites, certainly for the people interested in the evolution of it. We're studying invasive Phragmites as a standalone. We're not thinking about this whole other piece, which is the native Phragmites. And I think, I don't know, maybe I'll study the native Phragmites a little bit more.
Sounds great. Thank you. Would appreciate that. Anybody out there listening as well, there's obviously much more to learn, as always, which makes it interesting. But thank you so much. Really appreciate that presentation.